Okay, class, we're getting ready to talk about something very detailed in cell discussion. It has to do with cell division, known as mitosis. Also, I'll discuss briefly meiosis, and these will be discussed in separate videos. Mitosis is when cells divide equally, making two cells exactly alike. We could not live if our cells did not divide because we are constantly losing cells every day. And the only way we keep surviving is to replace those cells. We could not grow if we could not divide cells, because you may recall there are two ways to grow: hyperplasia and hypertrophy. Hypertrophy is an increase in the size of a cell, but cells can only get so big because of that surface to volume relationship problem we talked about. So most growth is due to cell division. So growth and repair and replacement deals with cell division. Now, understand this. Cell division, though, must be appropriate. It must divide when it needs to divide. I'm going to say that statement again. It must divide when it needs to divide. It needs to be under homeostasis. When cells divide when they should not divide, when they divide abnormally, when they just start dividing uncontrolled, that is what tumors are. That's what cancer is. So you can see that this subject is very important. Uncontrolled cell division are tumors, whether they be benign or whether they be malignant. So thus, there has to be a system in place in life to determine the control of cell division. And so this video here discusses what we call the cell divisional cycle. The cell divisional cycle is the cycle when cells divide. It's not the life cycle. This is not the cell life cycle. This is the cell divisional cycle. Let's take it to the big world. Let's say that a lady up to around 40 or whatever can have a kid. I mean, you can have it after that time. I'm an OBGYN, I can say that. But let's say we talked about her reproductive years only. Not her whole life when she becomes geriatric and so forth, but her reproductive years. Then that would only be one portion of her lifespan. So consider this. The cell divisional cycle is one portion of a cell's life. It is the portion of a cell's life when it can divide and is in the process of dividing, to put it more clearly. All right. So the cell divisional cycle is a series of events that take place in a cell leading to its division and duplication. In cells without a nucleus, prokaryotes would be like the bacteria, the cell cycle occurs via a process called fission. That means it just splits. But in the more advanced cells, the eukaryotic cells, it's a series of very complicated sequential events leading to the final process. The cell that's going to divide is called the mother cell. It has nothing to do with sex. It's just a generic term derived by someone, which meant the two new cells that become due to the split of the original cell are called your daughter cells. Right? So this is a picture. This is the original cell, let's say, called the mother cell. This cell did divide, so this cell is no longer there because this cell now are these two cells. When the cell first divides, now these are daughter cells, when it first divides, these are two small cells. They've got to eventually, each of them, grow back to that size. So then I've come to this cell divisional cycle. These are the sequence of events we'll talk about. But here are the two daughter cells that just were formed, And now I'm starting this cell divisional cycle, which is really a time clock. Until these two new cells, now each becoming a mother cell ultimately, then make their two daughter cells, and the process begins again. All right? So now we investigate into this cell divisional cycle. The cell divisional cycle is arranged, first of all, into what I'm going to call interphase and M period. Okay. Prior to the electron microscope and the more advanced light microscopes, you could not see much. Interphase was a period in which the genetic material could not be seen. If you look back at one of my earlier videos, the genetic material was in the loop domain stage, which meant it was beyond the resolution of the light microscope. Once mitosis occurs, the genetic material goes from the loop domain to the chromatid chromosome, terms you've heard. At that time, it is thick enough, it has been folded thick enough to be seen under the light microscope. So the only time under the older microscopes, light microscopes, that anything could be seen involving cell division was during mitosis or meiosis. Therefore, they called that the M period. The thought was that this was the real event. And then finally, you wouldn't see the genetic material anymore, and then it would come back. So they said, well, the interphase is a phase between the M period because you don't see anything. So it's just like they would say, okay, I see something, now I don't. Now I see something, now I don't. So there must be this little period between there. When I was taking the course years ago, they called that the resting period because we couldn't see anything. The resting period. Now we know, hey, the real workings are there. I want you to think about a stage play. I'm sure some of you have been in stage plays. A lot of you have seen them. The real working is not when the play is going on. It's all that practice before the play goes on. Because the play may only be 2 hours. You may have practiced for 6 months. So the real work was behind the scenes that you never saw. The people on the stage, you see them, but only for a couple of hours. So assume interphase, which used to not be able to be seen, is really where all the work is. This is where the trouble comes in cancer. This right here is just a period when you're actually carrying out the finality of the duty. So interphase, a period cycle that for years was felt to be a resting phase due to the fact that the genetic material could not be seen. It was beyond the light microscope. Interphase is broken up into a G1, a S, and a G2 period. Okay? The G1 period is called gap 1, the S is called synthesis, and the G2 is called the gap 2 period. Okay? Here you can see the genetic material is too thin to be seen. This is interphase. Here's again showing you the folding pattern that I showed you before, which meant that the genetic material is in the loop domain stage because this is the only time you can see it, which would occur during mitosis. Interphase, about 90% of the time, the cell is in the interphase, G1, S, G2. Okay, here we are right here. Okay. G1, S, G2, all in interphase. Here becomes mitosis there. The G1 time period is the variable time period. It's the period in the cell divisional cycle that can vary in its time span. Basically what happens in the G1 period is the growth period. Remember back here that when the cell divided, these two little cells were formed. Now what we need to do is grow them back to the size of a mother cell. So that G1 period primarily is a growth period. The other thing, when these cells first divided, they lost their differentiation. So also during the G1 period, the cell has to re-get its organelles and its function together for its differentiation, to become 2 kidney cells or 2 liver cells or whatever. So you can read there. Okay. So if the cell, though, here comes the next thing. If the cell is no longer going to divide immediately, it leaves the cell divisional cycle and goes into G0. Now let me explain what G0 is. G0 means it's no longer in the reproductive period. All right. Here's how this goes. I'm going to have to do a video 2 on this. So you'll have 2 videos on this that you'll look for. As long as the cell is running this cycle here, if it is a kidney cell, it's not doing kidney work at all. Think about, I'll just pick Macy's out. Macy's once a year or twice a year will close for inventory for a few hours. Now Macy's sells to the public, but when it closes to inventory, it's not selling to the public. It's consumed in its own internal operations. So it's not doing what its salesmanship is. When the cell is running this clock, if it's a kidney cell, it's doing no kidney work. If it's a liver cell, it's doing no liver work. The only time it can start functioning for its job is to leave the clock and to go into G0. It will go into G0 if it's not going to divide immediately, fairly immediately, or will never divide again. Nerve cells never divide after a certain age. So G0 is when a cell is doing its actual work. In cancer, for example, the cells keep running this clock and running this clock and running this clock. They never go into G0. So you will have a bunch of cells, but they do nothing. And as a matter of fact, they crowd out the good cells that are. That's what really kills you. When cancer invades, the cells start multiplying. So the person will have a bigger liver, but the liver cells aren't doing anything, or a bigger spleen or whatever. So G0 is an important time because that's when the cell is actually doing its work. Otherwise, it's running this clock. The S phase is when the DNA is synthesized. Now remember, if you split into 2 cells, you only had enough in the mother cell for one cell. So you had to have a time to split into the 2. Now remember, the G0 phase was the variable. I didn't put a time period. The S period, it takes about 10 to 12 hours to duplicate DNA. It takes about 10 to 12 hours to duplicate DNA. DNA duplication, I won't get into that heavily, basically involves a lot of enzymes and everything. But when DNA, you can read on this, duplicates, and here's kind of the thing, it will stay attached for a while. We'll be talking about when I get to the mitosis video, called a chromatid, 2 chromatids making a chromosome. The 2 chromatids actually are 2 strands of DNA, but for a while they stay attached. It's kind of like kid just born and he's still attached to the mom by the umbilical cord, let's say. All right. So the S phase is to duplicate the DNA. Here we are. All right. Then we come to the G2 period in interphase. That, in fact, is the period, that's the period in which, and it's only about 4 to 6 hours, that you're ultimately getting ready to do the stage play mitosis. So this would be like 30 minutes before the stage play is to go on. The stage director says, does everybody have their props? Everybody have their costumes? Because once we get started, we can't stop midway. Okay. So this, we're going to call this cell divisional cycle video 1. And then I'm going to do a 2 in a moment.